Hey there, my name is Dan Abrams, and I'm a clinical assistant professor in the Department of Psychiatry and Behavioral Sciences at Stanford University. And today I'm going to give a talk about some of our music research that we performed several years ago. And before I start, I'd like to thank Livia for inviting me to give this talk. Let's get started. So, who am I? And what gets me out of the bed in the morning? Overall, the goal of my research is to understand auditory brain function in humans with a focus on speech, voice, and music perception. And over the last several years, I've really focused on better understanding behavior and brain differences in clinical populations with auditory impairments like autism. So kids with autism have an interesting kind of auditory perceptual behaviors. They often tune out from the voices around them, and we think that this is an important component of their social communication differences and difficulties. And so the last several years of our work has focused on understanding brain differences in kids with autism with regards to voice and speech perception. But today we're going to talk about something different. But if you're interested in our autism research, please do look us up on PubMed or on our lab website, because we have all of our papers posted there, and you can feel free to click through and take a look at what we've been doing in the area of autism. So the big picture of my research interests is studying both the central auditory system, which is this complex set of brain regions, which kind of originates at the cochlea and in the inner ear, and ascends all the way up to cortex. And of course, this is a complex system in and of itself. But I'm also interested in how the auditory system interacts with cognitive brain networks. Characterizing cognitive brain networks, what are cognitive brain networks? Well, it's largely thought that cognition relies on the functioning of large-scale brain networks. So we've kind of moved past the idea that single brain regions are able to do important things in cognition. It's really now thought that it requires, cognition is reliant on the cooperative activity and connectivity between distributed brain regions, both cortical and subcortical. And so here, for example, is what's thought to be the semantic network. And you can see that, processing of semantics or identifying knowledge in various tasks in a scanner while measuring brain activity in humans relies on this extended brain network that you see kind of encompasses prefrontal regions here, temporal regions here, parietal regions here. And so it's thought that these kind of higher level cognition is reliant on these kinds of networks. And so the overall questions that we're interested in, that I'm interested in particular, is how does the complex constellation of auditory brain structures work in harmony to facilitate biologically important human functions, including speech, language, and music processing. And so a quick and important question is, how do we study brain function? Well, of course, there are lots of ways of studying brain function. And some people, some labs and some groups, study relatively small and narrow areas of neural function, including how single neurons in the brain respond to different stimuli. And then, of course, you can get down to an even more detailed level to look at how, I guess it would be less detailed, but to examine how two different neurons interact at a synapse. And then, see, these are kind of micro level neural processes, because of course the brain consists of millions and millions of neurons and trillions or at least a trillion synapses. And then other ways to study the brain is to examine small populations of neurons. So you might consider this 5 to 100 neurons. And then larger populations of neurons. And so when we're talking about cognitive brain networks, we're talking about how millions of neurons function simultaneously across large swaths of the brain. And as you might kind of have a sense, how do we study brain function? Well, we study these large-scale brain networks, and it's kind of the highest level of of understanding brain function in the sense that we're looking at very large populations of neurons and how they respond simultaneously. We're not going to be able to track down how a single neuron responds using the methods that we use, but we can understand how brain systems, kind of large-scale brain systems, respond and to different stimuli and are associated with various cognitive functions. And to do this, we use functional magnetic resonance imaging. And this is a non-invasive method, which some of you, I assume, have some knowledge about. And the idea with this method is you lie someone down in a scanner, and you, in the case of kind of auditory stimuli and music processing, for example, you have them listen to various carefully chosen auditory stimuli, such as a speech stimulus or music stimuli, and you measure oxygenated blood flow throughout their brain, and we're able to, in the analysis, identify what parts of the brain become active in response to particular auditory stimuli. And so our general question is, how do large-scale brain networks facilitate complex cognitive functions? And in our case, in the study that I'm going to be talking about today, we're going to examine naturalistic music processing. And so what do we know about brain processing of naturalistic music? Well, most of what we know about brain processing of music in general is associated with how does the brain process particular components of music. And so, so we're scientists. Music is this big, complex kind of category of stimuli that involves multiple components. You have pitch, you have rhythm, you have timbre, there's harmony. There are lots of components of music, and they all kind of vary over time and during kind of natural pieces of music. And as scientists, our job is often to reduce a complex entity like music into its components, and then in the case of brain science, to understand how each of these components individually are, kind of individually represented in the brain. So, for example, one very important component of music is pitch. And so there have been lots of studies examining how the brain processes pitch. And so in a study like this, you'll have a musical tone at one pitch, and there'll be, I guess this is a spectral versus temporal comparison, but nonetheless, these are relatively simple stimuli used to examine these individual components of music. And what you can see here, for example, is that right hemisphere processing, right hemisphere cortex, auditory regions are particularly active for pitch processing. But again, they did not use a complex and natural piece of music to examine this question. They used a very reduced stimulus set to examine these very particular components of music. And a similar example is to look at tonality and rhythmic components of music. These kind of beat versus a no beat experiments are associated with this network here, which is kind of a cortical and basal ganglia network. And so, more example, we also have Tambor processing. This is another important component of music. What instrument is playing? Is it a violin? Is it a cello? Is it a viola? And this is from Vinod Menon, one of my collaborators. And he showed that bilateral auditory cortex underlies the processing of timbre in music. And so, while all this is very satisfying and interesting to know about these individual components of music, they really can't tell us what happens in the brain during a naturalistic musical experience. And it leaves us somewhat kind of, prior to our study, there really wasn't much known about the processing of naturalistic music. And so after giving this some thought, we realized that there was a method out there that was actually developed using for another study of visual processing that we could use to apply to understand naturalistic music processing. And the method that we used is called an intersubject brain correlation. What is an intersubject brain correlation? Well, the seminal study that used intersubject brain correlations was this really clever study performed by Yuri Hassan, who's now at Princeton. And what he did was he had viewers watch a movie in the scanner. And I don't know if you guys are familiar with this. This is probably a little old for you guys, but this is Clint Eastwood, and this is a very famous movie called The Good, the Bad, and the Ugly. And it's a famous Western film. And he had people watch this, again, it's a very uncontrolled stimulus. It's a movie. There are a lot of things happening. There are different people. There are different scenes. And as scientists, this is usually too uncontrolled for the preferences of most scientists, but here they did this ambitious thing, and they measured brain responses to this naturalistic, I'm going to quote naturalistic, viewing experience, which is this movie. And what they found is that, even though people's brains are quite different, and people, in the sense that we all have different experiences, we were raised by different parents, and we may have been raised in the Western society, but there are lots of differences and idiosyncrasies between our, our knowledge base and our interests. Nevertheless, they showed that different people's brains tick very much the same while watching a movie. And they used this brain correlation method, and here's kind of a map. This is kind of a strange map, but a flattened map of the cortex, of human cortex. Here's kind of a more natural view side view of the brain. This being the front of the brain, this being the back of the brain. And all these colors here, orange colors, represent are kind of statistical maps from their data analysis that show, well, what parts of the brain are correlated between subjects while watching a movie. And with these brighter yellow colors showing the greatest amount of correlation between subjects while they watch the movie. And what they show is that while watching this extended stimulus, the movie is a couple hours long, and that people's brains respond in a very similar fashion to these extended stimuli. And so we thought, wow, maybe we can apply the same method to studying a naturalistic musical experience. And so that's what we did. And here's a visual display of the stimuli that we used for our study. What we did was we took a, a symphonic piece of music by a composer named Boyce. Uh-oh, this isn't in revision. This was published in 2013. Sorry about that. And here's a time frequency map, a spectrogram of our naturalistic music stimulus. And as you can see, the stimulus is, I think it's about, how long is it? Oh, I should know this off the top of my head. These are in seconds, so I guess this is one of the movements from the symphony, and it's a couple minutes long, but there were several movements that we used. Anyway, and so here's the natural music condition, and you can see it varies. Here's some interesting spectral patterns that you see over time. And then we had two control conditions. We had a phase scrambled control condition, and, oh wait, let me see if I can play you the naturalistic condition. So, right, so this is a lesser known composer from the Baroque period. So we wanted to choose a piece of music that was going to be unfamiliar to our listeners. And so here's one of our control conditions. It's a phase scrambled condition. Basically what we did was we took this natural music condition and we altered the phase of it. So what this does is it leaves all of the spectral components intact, but it rearranges all of the temporal components of the music. And so all the timing information has been removed or reordered from the natural music condition while leaving the spectral components the same. So this is what it sounds. This is the phase scrambled condition. All right, you get the point there. And so we also had another condition where we rearranged. And now I'm going to show you some pictures of the brain. And in these pictures, you're going to see, obviously, here's a representation of a human brain, and also these colors on top, similar to the previous slide that I showed you from the Good, the Bad, and the Ugly study. These colors on the brain are statistical maps from our analysis, and if you see a color here, it means that it met our threshold, statistical threshold, and it means that these parts of the brain were significantly activated or were significantly correlated among listeners for a given condition. So here's a natural music condition, and the first thing I'm going to point out is that the natural music condition elicited synchronized activity in low-level parts of the auditory system. So this is the inferior colliculus. This is the midbrain, primary kind of midbrain structure in the auditory system, and it shows that this result shows that natural music, not surprisingly, drives synchronous brain activity across participants in this low-level auditory region. However, the phase scramble condition, the one without the temporal information in it, does not drive synchronized neural activity. And when you look at the direct comparison between natural music and phase scrambled, you can see that there are significant differences both in the inferior colliculus, which is the midbrain auditory structure, and the medial geniculate, which is the primary thalamic structure in the auditory system. And you can see that what this result tells us is that without the temporal information in the musical stimulus, there's no synchronized activity or significantly reduced synchronized activity in the inferior colliculus and the medial geniculate relative to having that temporal information in natural music. And I guess from one perspective, this isn't that big a surprise. We know that auditory neurons very much are highly sensitive to timing information and fire robustly in response to super threshold and to loud enough sounds with very kind of clear temporal information in it. And so when music lacks that temporal information, it kind of is quite plausible that these low-level auditory regions are fail to be synchronized between participants. Okay, moving on though, what we found is that natural music also drives synchronized activity well beyond these kind of low-level subcortical auditory structures, but also drives synchronized activity in primary auditory cortex. So now I'm going to show you brain maps. Here's the front of the brain, here's the back of the brain, and we've outlined particular parts of the brain that are different structures of auditory cortex. And so here's HG, Heschel's gyrus, which is thought to be primary auditory cortex. This is the first stop in the cortex for auditory information processing as it ascends from the ear. And then here are two nearby auditory cortical regions. PT is planum temporale, and here is posterior superior temporal gyrus, another auditory sensitive region. And we've outlined them, we've done our best here to outline them here, and then you can see overlaid on those outlines are our statistical map, and you can see that natural music drives highly correlated brain activity between participants in these low-level auditory cortical regions. That includes planum temporale, Heschel's gyrus, and posterior superior temporal gyrus. And you can see that without the temporal information in the music, in our phase scrambled condition, you can see there is significantly reduced brain synchronization in these low-level auditory regions. And when you do the direct comparison between the natural music and the phase scrambled condition, lo and behold, it shows that there is significantly greater intersubject brain correlation for the natural music compared to the phase scrambled condition. Once again, highlighting the need for temporal information in music in order to drive and synchronize brain responses in these auditory cortical regions. So what was interesting is that these synchronized brain responses weren't just restricted to these auditory regions, either the subcortical ones that I showed previously in the inferior colliculus and medial geniculate, also in these auditory cortical regions, but we also showed that intersubject brain synchronization for natural music was also evident well beyond these auditory structures in the brain, and started to synchronize other brain systems. And so now I'm showing you a slide. Here is a coronal view of the brain, and this is a prefrontal region. These are Brodmann areas 45 and 47. I guess these are kind of more frequently thought of as Broca's area in prefrontal cortex here. This is right hemisphere, though. Broca's is typically associated with left hemisphere prefrontal regions. And now we're also showing a coronal slice of parietal cortex. And this is PGA is a region of the angular gyrus, and SPL is the superior parietal lobule. And what you can see is that natural music drives synchronized activity also in these prefrontal and parietal regions. Now these regions are not, of course, just selective for auditory stimuli, unlike the previous regions that we showed back here in auditory cortex. However, these regions are considered heteromodal cortex. This means that all sorts of different stimuli activate and are, these brain regions process all different kinds of stimuli, including visual stimuli, auditory stimuli. They're associated with, these prefrontal regions are associated with complex cognitive tasks such as cognitive control. And so these are well beyond kind of the canonical auditory system. Nevertheless, natural music just music stimuli that we hear actually drives these heteromodal brain regions of prefrontal and parietal cortex, and it drives synchronization in these regions across participants. And this is less so of the case in the phase scramble condition, which has the reduced and kind of temporally reordered stimuli. And again, in the direct comparison between natural music and phase scrambled, you see that there are these significant differences in these prefrontal and parietal regions. And I guess our hypothesis here is these regions are often associated with attention, auditory attention, and so these regions may well be helping and facilitating participants to remain engaged with the stimuli over extended periods of time. In addition, they could well be important for integrating auditory information over long periods of time. It's thought that understanding and kind of comprehending and putting together a musical information over extended periods of time requires us to maintain this music in working memory, and also to enable the kind of integration of this information over time. Oh yeah, I remember this theme in the music. It came up in the first 30 seconds of the music, and here we are 2 minutes later. I recall this theme, and the brain systems that may help us keep this theme in working memory over this period of time are likely, at least partially, associated with these prefrontal and parietal brain regions. And taking us down the home stretch here, we also found motor regions are significantly correlated between listeners during a natural music experience. This is premotor cortex, and this is the MCC. This is cingulate cortex here, and it's kind of middle cingulate cortex, or medial cingulate cortex, sorry. And these are kind of important motor processing regions. These regions are appear to be kind of less activated or less synchronized during the phase scramble condition. And when you do the direct comparison between the natural music and phase scramble condition, you can see that there are these significant differences in these motor processing regions. And so why in the heck would you see motor regions during a musical stimulus? Obviously, there's no motor task that we're asking the participants to do. Well, there are important links between auditory and motor processing regions. When it's thought that oftentimes when you have participants do a speech perception task, it will activate these speech motor cortex. The reason for that is kind of hypothesis for that is that that as we hear speech, we mimic some of those speech sounds in our brains and in our minds. The act of making those sounds that we're hearing, and there's this kind of important and maybe kind of obligatory connection between these auditory processing regions and these motor preparation and brain regions and premotor cortex. In the case of music, it may well be the case, you hear this beat, often your body wants to move in response to the beat, or there's this kind of there's this rhythm in the music, and our motor regions often want to synchronize with those rhythms that we're hearing. And this is all kind of conjecture, hypothesis here, but our hypothesis would be that these kind of motor regions associated with rhythmic processing are synchronized between participants as a result of the rhythmic information. And you can see, and this is evident in both premotor and cingulate cortex here. And so, what have we learned about brain processing of naturalistic music? Well, what we learned is that a widespread brain network tracks musical structure across long periods of time. And, one of the things that we showed is that, during these naturalistic music, despite our idiosyncrasies. These people had never heard this voice music before, this symphonic music, this particular symphonic music before. People may not really like symphonic music. People have different musical interests. And despite everyone's kind of different relationships with music and different experiences with music, people showed very similar brain responses to this very extended and naturalistic musical experience. And that this widespread brain network, which extended well beyond auditory processing regions, but also into memory, working memory, attention, and motor processing regions, were also synchronized across participants in response to these sounds. Moreover, kind of mimicking what I just said, these extended networks is served by brainstem, auditory cortex, attentional networks, and motor planning regions. And an interesting question is, how can we extend this work? Of course, we can. Interesting questions are associated with clinical populations in which auditory integration that may have auditory integration disorders. For example, some kids with learning disabilities and some kids with autism may have difficulties integrating auditory information over extended periods of time. One way to think about this is, if you had a kid listen to an extended narrative told by another person, and the goal of the narrative is you have to kind of keep track of all the information that's been told over several minutes. Different populations. It would be interesting to see what part of the brain and what part of is having trouble synchronizing, and during a naturalistic and extended narrative that a kid might hear in a more kind of naturalistic social environment. And so, and of course, another way to extend this work is to examine the processing of key components of musical structure using naturalistic stimuli. Like, for example, what are their key components? Here we kind of showed in a broad way, we showed that kind of temporal structure and is important. Once you remove the temporal structure of the music, a lot of the brain synchronization was reduced in these brain regions that included auditory, attention, memory, and motor processing regions. But what are the other kind of key components of the musical structure that were necessary to drive that brain synchronization? And so that's all I have for you today. I always want to acknowledge my awesome collaborators at the end of my talks, and of course, our funding sources who funded this work, including NIMH and Brain and Behavior Research Foundation and Safari. And thank you very much for your attention. If you have any questions, please feel free to email me. You can reach me at daa@stanford.edu. And thanks again for this opportunity. The temporal components, we rearranged the spectral components while leaving the temporal components intact. And I guess I don't have I think the stimuli, you can find that stimulus, sorry, it's not here, but you can find it on our website if you're interested in hearing what the spectrally rotated or reordered condition sounded like. It was a second control condition. And what we did was we had each participant come in and have their brain scanned while listening to each of the three stimulus conditions: the natural music condition, the phase scrambled condition, and also the spectrally reordered condition. Sorry about that. And in the analysis, we examined brain differences between these musical contrasts using this kind of intersubject brain analysis. Studies, they've shown that these kind of prefrontal regions are associated with the processing of tonality,